Okay, I think we can start. So welcome everybody. It's my great pleasure to introduce Bogdan, who is a computational biologist. In fact, sparing his blushes, I think one of the most inventive computational biologists that I know. His background has been in genetics, but as you'll hear from today's talk, as is true, I think, of most of the computational biologists, they're beginning to take advantage of additional sources of high-dimensional data, in this case, the electronic health records. And my intention in inviting Bogdan was for him to introduce you to some of the resources that have been developed at UCLA, which I suspect many of you will not be familiar with, and to give you an introduction to what those resources, these clinical resources, can do, particularly when combined with genetic data. But I should emphasize, not just with genetic data. There's a lot of interesting questions you can raise with these data on their own. And do regard Bogdan as a portal, as a way of getting access to this, and for you to getting help in asking questions which would be relevant to your clinical practice. And just one note about the seminar: we'd like you to use the Q&A box to ask questions rather than enter them into the chat. So that should be at the bottom of your screen, where it says Q&A. So I'll hand over now to Bogdan, and for the rest of you, please enjoy his seminar. Thanks, Bogdan. Bogdan, you're muted at the moment.
There you go. Too many screens. Thank you so much, Jonathan. Hopefully everything works out now.
I'm sure it will. Okay.
Right now. Thank you so much for the lovely introduction and for allowing me to speak to all of you. I'm particularly excited to present to you by way of introduction of some of the resources that we've been building here at UCLA. And by we, it's a very global we that you'll see, anywhere from the Institute of Precision Health to computational medicine to the CTSI, and hopefully some of you in the audience that are doing critical work for developing these resources, and hopefully they will prove useful for your research, and clinical in the future. So today I'll be talking to you about some of the resources here at UCLA, and I'll give you some of the examples that we've been using in our own research of how these types of data can be used to answer interesting questions at the realms of putting genomics at the forefront of clinical practice in the long run. So I'll start by very briefly give you an update, if you will, a two-slide update of where genomics currently is, and then go through some of these resources that I hope they'll prove useful to you. And I'll point you to different places where these resources can be accessed, and we can go from there. So before I get started, I should acknowledge the key people that have made a lot of contributions to all the work that I'll be presenting to you today, from my group particularly. And these are grad students from a variety of disciplines. So Malika Front, who's currently training to be a genetic counselor, Ruthie Johnson, a computer science graduate student, Tomer Schwartz and E. Ding, who are PhD students in the bioinformatics PhD program. So one of the things that you'll see throughout the talk is the interdisciplinary component of all the research that gets done. So what do we think about genomics and how that works with phenotypes? So broadly speaking, we can distinguish among two classes of traits, or two classes of human diseases. We can distinguish among diseases that are Mendelian phenotypes, so things that are typically large effect, where genetics contributes tremendously to the phenotype. They're typically single disruptions in key genes. They're typically rare because of that, so they're high effect. And in general, genetics, contributes, it dominates with respect to how that particular patient will get that disease if they have those disruptions. So in the environmental, it's typically small magnitude there. In contrast to this Mendelian phenotypes, or that have been traditionally referred to as genetic traits, if you will. Most of the other phenotypes that we're typically looking at that are common in the population, they tend to be complex, and they're complex on different dimensions. One first one is that there's typically many genes that contribute to these traits. These genes will have tiny little effects, but it will aggregate across many genes that hit critical pathways at different time points in the evolution of the disease. And it's typically the case that there's a strong environmental or non-genetic, non-inherited contribution. And that varies across different traits. But the first approximation can imagine genetics, contributing roughly half, and then the other half would be kind of non-genetic contribution like diet, exercise, and so on and so forth. So if we think about these two broad classes of diseases, the genomic revolution has been particularly successful for Mendelian phenotypes, to the point of, in the past decade or 15 years, we've gone from discovering or understanding the genetic basis of these phenotypes all the way to clinical practice. So, and I'm showing you here some very or a few examples from UCLA. UCLA has been a key player in this domain, by which currently all the clinicians can order an exome sequencing test that will search through the exome of a given patient that might be suspected to have one of this Mendelian severe phenotype, and we'll go through the process of actually assigning of diagnosis based on the genetic variants that this particular patient might have. And to the extent that we currently have at UCLA California Center for Rare Diseases, this is through many work of many people here at UCLA, such as Stan Nelson and his team and many others. And in many ways, that's the promise of the genomic revolution to clinical practice. In contrast to that, for complex phenotypes, we're not there yet, and presumably we won't be there yet for the foreseeable future. And you can argue that because environment contributes way more to these phenotypes, the way that the genomic revolution will go through the clinical practice for complex phenotypes would be of different nature than what's happening for Mendelian traits. So what I'm showing here on the right is just one example of how over time, which is on the x-axis there, we keep on cataloging more and more regions in the genome that harbor variants that contribute to a variety of complex or common diseases, anywhere from cancers to brain-related traits to autoimmune traits. And we see that over time, we keep on accumulating all this knowledge of regions that contribute or provide risk to particular individuals for developing those disorders. The challenges comes from the fact that all these risk regions typically contribute a tiny amount, and so the chances of a particular patient to get that disorder. And in aggregate, very few patients or very few individuals have, all the risk variants for these thousands of regions in the genome, such that they would have a high probability of getting the disease. So we're not there in terms of utilizing these genetic findings as of yet towards clinical practice for the genomic for complex traits. So within this 2-slide summary of where we are in terms of genomics for understanding disease and moving it towards translational practice, a new type of revolution is kind of happening in terms of how do we collect data for our patients. So if we think about 5 years from now, we would want to take all these discoveries and somehow implement them and do something better, some better clinical outcomes or improve clinical outcomes for our patients. The first thing that we would need is genetic or genomic data for our patients. And this has been kind of a new wave of types of data that have been generated sooner for some places than others. Here I'm showing you the year that enrollment started, by which a lot of medical systems have created biobanks by which patient data, or genomics in this case, but in general, there's data, or it's being biobanked, samples are being biobanked from the patients that are within the health system. And that's kind of a foundation for where, implementing anything that we might want to do in terms of genomic medicine. Here in this plot, I'm showing you where UCLA stands. So you see that right there on the right side, UCLA Precision Health Biobank, which I'm going to describe to you a little bit more thoroughly today. And you see it in contrast with other types of biobanks that are existing within the world. And on the y-axis, it's just a measure of diversity, and that's measured in this case as a percent of non-European ancestry participants, so individuals of non-ancestry descent. And you would see that UCLA has quite a diversity, quite an amount of diversity, as we all know and appreciate. But more importantly, we cannot really, unless we generate data and on our patients, we cannot really think about translating all the other discoveries from all the other biobanks or the other data sets towards our patient population. So with that in mind, today I'm going to talk to you about two big resources that have been put together by a lot of individuals, some of them hopefully on this call, and they're kind of managed by the Institute of Precision Health here at UCLA. So a one-stop shop for those of you that are interested in these resources would be to go to this website of the Institute of Precision Health, and there'll be a link of exactly more thorough description of this resources that I'm presenting for you today, as well as a way by which everyone at UCLA can access them and can utilize them in their own research setting. So one of the main resources here would be the data discovery resource. And you can think about it as a de-identified version of all the electronic health records at UCLA that have been put together after stripping of all the identifiers primarily for research purposes. So as probably all of you are fully aware, UCLA is serving a wide range of patients, and starting from 2013 or so, when the electronic medical records were implemented at UCLA, we have a unified version of all the electronic health records that at this point, by some counting, are about 5 million patient records over time. Obviously, not all these patients are active within our system. Again, these will be patients that have been within our system at any point in time in the past 6-7 years. And because of the de-identification project is ongoing, we continue to add a lot of resources to it. So at this point, you can imagine that this data discovery resource contains a lot of billing codes, all the lab values, medications, demographics, and so on and so forth, with ideas about adding imaging data to them and adding de-identified nodes, and so on and so forth. So to the extent that this is a resource that it's highly useful for the community, more and more types of data modalities will be added to it. And in addition to this, because I was talking about genomics, the other flagship resource that the Institute of Precision Health here at UCLA is creating is an atlas of genomic variation of all of our patients. And this has been aptly named as Atlas Community Health Initiative. I do not know what Atlas stands for, so if there's a question about that, I'll defer to the Institute of Precision Health, what's the acronym or the backronym for it. The main goal of this flagship project for the IPH is to collect data from 150,000 of UCLA patients. So patients have, as they go through the UCLA clinics, they are being provided the opportunity for universal consent for de-identified research. Essentially, there are a bunch of iPads that patients get to see. They watch a video in the language of their choice, and then they consent, if they want to, they consent about whether their remnant data would be used in a de-identified manner for research purposes. And then the next time that they come to the hospital or to the clinic, an extra vial of blood will be collected from them. And with that biobank type of data, the first initial wave of data generation from them for these patients would be to collect what is called genome-wide genotyping arrays. So you can imagine this as collecting the common genetic variation, in the population at around 500,000 to 600 sites in the genome. This is, this data has gone through layers of what is called genotype imputation. So this is computationally filling in the blanks to get from 500,000 or 650,000 variants to about 10 million variants across the genome to capture, broadly speaking, most of the common variation that or genetic variation that these individuals may have. So this gives a very comprehensive picture of variants that exist in our patients that are common in the sense of at least 1% of individuals might carry those mutations. And just to give you a very very high view overlook of how this consenting and how this biobank is progressing. Here on the left side, you see all the sites or the clinics where consenting is ongoing and or at least was ongoing as of pre-COVID times. At this point, it's a little bit on pause, as you might expect, and you see there all the sites, and one of them is including in the Semel. So thank you for all of you that are participating and contributing to this. And on the right side, you see, and I'm not gonna click on it. It's a 5 to 10 minute short video that can appear in multiple languages where patients get to watch and then consent. So as of a couple of months ago, about 77,000 patients completed the consent. About 50,000 opted in, and from those 50,000 patients that opted in, about 35,000, a little bit more than 35,000 got their specimen data biobank and de-identified, and from them around 35,000 have been already genome-wide genotyped. And some of the data that I'm going to show to you today, it refers to this first wave of data that's available in a de-identified manner about these 35,000 patients here at UCLA. So in summary, the grand picture of these two data sets are as follows. There's the de-identified data resource, or the DDR, that contains a lot of the phenotype information, a lot of information that one might want to use to ask research questions, and they're linked to the genotypes. And I should say that this is an ongoing process. Most of you probably have seen the announcement that Institut Prestigieux has struck a deal to actually provide sequencing in addition to the genotyping. So all these individuals that are being, or these patients that are being consenting and data has been generated, they will be whole exome sequence through partnership with Regeneron. And as Jonathan mentioned at the beginning of this talk, this is an ongoing resource with more and more data that will be added to them, again, for the benefit of all UCLA researcher and beyond. So how does the data look like? Well, as you probably are fully aware and fully appreciate, we serve a wide range and diverse set of patients here at UCLA. This is just a snapshot of how the self-reported race ethnicity looks like. And you see that we serve a wide range of individuals of various and diverse race ethnicity groups. Obviously, this is just one way that we can look at diversity, and we can pull lots of data in a de-identified manner about other types of referring to that diverse. And in terms of genotyping, we're genotyping roughly similar proportion of diverse individuals. So that's kind of one of the the desires of our genotyping, or all this process, is to not bias versus one versus other types of individuals in the data. The only thing that you will see here, you see that on the orange, on the second bar there are individuals or patients that don't identify as any race, ethnicity in this case. And then those patients are, essentially most of the time they don't want to participate in this assay or in this consenting. So those patients, they don't get to be genotyped, but everyone else, you see wide representation across all diversity groups. So we're capturing a wide range of individuals of very diverse backgrounds. So now that we are putting this research together, one of the first questions that we're always left with is, how do we provide a quick and fast, or a fast and accurate, I should say, way of extracting phenotypes from the electronic health records? As most of you are fully appreciative, I suspect, electronic health records are very complex, and they're very biased and noisy and so on. So a way to extract meaningful information out of it is typically desired for any of these resources to be useful in practice. And one of the main way that we and others have done this types of data extraction at scale is by billing codes. So this is probably the simplest and the easiest way that we can, pull information about how many patients do we have that have a particular ICD code. And in general, they're the closest approximation for phenotypes. And they're used through multiple medical institutions, internationals, and we could essentially use this to aggregate data, if you will, across multiple sites. The challenge is that the ICD codes are not really designed for phenotyping. They're using for billing, which comes with a lot of biases. Most of the times they're so well detailed, and I'll show you in the next slide what I mean by that, that it's typically not useful to do research or to extract data just based on the billing codes. One would need extra information for the traits that are desired to do more than just the ICD. And probably for research purposes, they're not really aggregated across a clinical representation of diseases. So they mix together diagnosis with history of disease. So it's kind of a mess, if you will. And they're not diagnoses, but symptoms, place of occurrence, and so on and so forth. So it's the fact that they are using for billing comes with a bunch of drawbacks that we, for research purposes, we want to overcome. And just to give you an example of one type of billing code that here, given that we're close to the ocean, we might encounter. Here's some example of billing codes that deal with contact of a patient or potential patient with marine animals. So here there are about 20 billing codes that deal with whether that particular patient was struck or had a contact with dolphin or sea lion. They're further, broken down by whether it's bitten, struck, or other types of contact, or whatever that may mean. And even within those categories, we can see billing codes that, ICD codes that deal with whether it's the first time that somebody was in contact with a dolphin, second time, and so on and so forth. So just based on this, you can realize that it's this very detailed granularity. It's not really meaningful in general for research or genomic research. And if you thought that sea lion and dolphin are the most important marine creatures, well, there's orca as well that can be contacted, and that's put in its own billing code. And unfortunately for all the other marine animals, they're clumped together in the other categories. But as you might expect, these are just the mammals. Here's other billing codes that deal with sharks, that deal with fish, and so on and so forth. And just from looking at this complexity, you can realize that here's about 30-50 billing codes that deal with something that may not be a useful phenotype for general research purposes. So what we've adopted here on top of the DDR is to use a grouping of ICD codes that is referred as Phecodes. And this has been proposed by our colleagues at Vanderbilt almost 8-9 years ago. You can find it in this PWAS catalog nomenclature that takes all this ICD 9 and 10 code, about 60,000-70,000 of them, and groups them into aggregation of billing codes. Sometimes they're negations as well, so they have to have a set of ICDs, but not others, to create more meaningful representation of diseases and traits. So from 70,000 ICD codes, we get into around 1,800 Phecodes, and these Phecodes are further stratified in different groupings that are more biologically meaningful. So how are these 1,800 Phecodes represented here at UCLA? Here's a distribution of this 18 classes of Phecodes, anywhere from, let's say, circulatory system, mental disorders, all the way to this general category of symptoms. And here on the x-axis, you see the number of patients in our data in the DDR that have a Phecode in this category. So you see that, let's say, for mental disorders, about 500,000 patients here at UCLA have a Phecode that it's within this category of mental disorders. And because this DDR is a temporal resource, we can also think about the number of diagnosis events, defined here as the number of distinct Phecodes that a particular patient might have, distinct in the sense of like at different time points that a particular patient might have. And looking at, let's say, mental disorders as an example, 500,000 patients, and then maybe a million diagnosis events. So roughly 2 to 3 diagnosis events per patient if you want to do quick stratification, or if you quick aggregation of data. So because we're interested in, or because the audience is mostly interested in mental disorders, we're at Semel here. Here's a breakdown of, in terms of thousands of patients on the x-axis, see it on the top, of the Phecodes within the mental disorders category. So remember, there were 500,000 or so patient records here at UCLA that had a Phecode within this category. There are 77 types of Phecodes within the mental disorder category, that grouping. And you can see them broken down by, or rank ordered by, the number of patients that have at least one occurrence of these Phecodes in the UCLA patient data. So you see anxiety disorder comes on top. About 200,000 patients have at least one Phecode from that disorder. Mood disorder, major depressive disorder, it's about 170,000 patients at UCLA that have a Phecode for that, and keeps on going down. Alcoholism is around 30,000, psychosis 10-15,000, dementias 20-30,000, and so on and so forth. Or autism, around 10,000 patients here at UCLA. So just by this quick overview, we're using Phecodes, you can get a very accurate, if you will, snapshot about the types of patient records that we might have access to them for the purpose of performing, let's say, research or querying this data for different types of projects that we might have. And these are just the top Phecodes based on that have more than 10,000 patients. We can look at the ones that are under 10,000 patients here at UCLA, and that's there you find 8,000-9,000 patients with Alzheimer's, schizophrenia, conduct disorders, and so on and so forth, all the way to dementia and so on and so forth. So this very quick resource can allow you to interrogate this data quite fast, and you can see in this case I'm just showing you the breakdown, but obviously you can ask questions about what are the co-occurrences of different traits and whether one trait occurs before another, and so on and so forth. And the data is large enough, whereas for most reasonable queries, you might have enough patient records to actually answer those questions. Getting back to the broad categories, we looked briefly across how they stratify across sex and age. And we've done this more or less as a quality control to make sure that everything looks fine. And as you would expect, we see things that are that make sense. We see congenital anomalies are overly enriched in younger patients than overall, and or that genitourinary traits or Phecodes are enriched in women, and so on and so forth. So this is more kind of like a snapshot of that the data is high quality. We also looked at by partitioning it by self-reported race ethnicity, and there we already see things that might be interesting from a research perspective as a potential investigative tool. For example, we see pregnancy complications are overly enriched in non-European ancestry individuals. So all these things are already could be mined for different research questions, and I'm inviting all of you to think about whether this type of research could be useful for your potential interesting questions. So to do a more thorough job about quality control here, to make sure that we're not really observing anything that we're not supposed to observe, we took a bunch of example traits for which we knew what we were expecting, and we used them as overrepresentation to make sure that the data is correct. For example, we looked at Alzheimer's disease and asked that whether individuals that have a Phecode for Alzheimer's disease, whether they are over 65 years old or not. And in the DDR, as I showed you before, there's about 7,400 of individuals that have a Phecode for Alzheimer's disease, and 6,800 of them are over 65 years old. So by any measure of significance, that's super highly significant. So indeed, this serves that the data is of a good quality. Similar with sickle cell anemia, where we observe a massive enrichment within the African American self-reported individuals here at UCLA, again, as we expected. Looking at autism, we see what we all know, that the male there's an enrichment within male individuals. In this case, about 5,000 of individuals that have autism or Phecode of autism here at UCLA and the DDR are male, and that's the vast majority of all the individuals that have a Phecode of autism here at UCLA. Same thing with multiple sclerosis, and then now we can automate these queries, and then we can search these types of enrichments across any type of pairs of traits that you might want to be interested in. For example, we are looking at spina bifida, which is a disease that it's kind of rare, and we see a massive enrichment within, or depletion, I should say, within individuals that self-report as white or Caucasian. And interestingly, this has been postulated and presented in some literature. So this is, if you will, a snapshot of the data that exists, and hopefully that gives you guys some ideas on how you can utilize it for your own research and beyond. So the next part, or the second part of my talk, I'm going to briefly present some examples of research that we've done on top of this data, but as a way to give you ideas on how we can leverage this type of resource for our own benefit. So in the first vignette of what can be done with it, we can take one genetic variant and we can ask whether that individuals that carry that genetic variant that have a particular disorder, whether they also carry other disorders. And I'm showing you here through an example what it's called, this technique is called PheWAS. So I'm showing you through an example of IRF4, which is a well-known gene for skin cancer, risk for skin cancer. And what we asked here is whether individuals that carry the risk variant for IRF4, whether they have other types of traits in our biobank, in our EHR. And what you observe here on the x-axis, there are these 18 classes of different traits that are in this Phecode database. On the y-axis, it's the strength of the association in units of p-value. So whatever it's about that threshold, that line, it's significant. Everything else below, it's not significant. And as you might expect, any type of skin cancer, either non-epithelial, squamous cell carcinoma, basal cell carcinoma, they show up as highly enriched. So patients that have genetic variants within this IRF4 also are enriched for these other traits. And as you might, interestingly, we also see actinic keratosis, which is another skin disorder or skin trait, which is highly related to skin cancer, or if you will. So this type of data where we have genetic data as well as lined up with the electronic health record or information from the electronic health record can allow us to quickly search for potential function or of particular genetic variant. In contrast, we can also do the reverse, where we can focus on one particular trait and we can march along the genome and ask whether those particular variants are correlated with a trait. I'm showing you an example from a trait like gout, and there we see similarly as what's been reported before, regions in the genome that are correlated with, in this case, gout in Europeans. And these are all examples, snapshots of the things that one might be able to do with this type of data. And obviously, we're not operating in a vacuum. Many other places are creating their own biobanks, and we've partnered with this 20 other biobanks worldwide, by which we share our information about our findings, such that by increasing the sample size, we have a better chance of discovering novel genetic variants that are driving different traits. Here's a snapshot of one of the most recent type of aggregation of this type of data. Here's 15 example traits from asthma all the way to appendectomy and others. And what you see here on the y-axis is the number of regions in the genome that are significantly associated after pulling together information from these 20 or so biobanks. And in the red, there are the regions that have not been reported before. So this showcases that by working together with other biobanks, by increasing sample sizes, we can identify a lot of new risk regions for all these traits. So this showcases that this is probably the next wave of how genomics will get done in terms of discovery. So another vignette of how we can utilize this genetic data for doing a better job of in the clinic is in in curating which variants might actually cause disease and might not cause disease. And this has been reported a couple of years ago in the seminal paper from Man Rai et al., where they started with a very simple question. So they looked at a trait, cardiomyopathies, that are diagnosed based on mutations in 5 or so genes. And what they observed, their observation was that after the diagnosis, they observed that African American individuals tend to have a higher incidence rate. So based on the diagnosis after performing genetic testing. And what they noted, and this is what's shown there on the right, is that after looking at these mutations that are used for genetic testing, these mutations are overly enriched in African Americans over in individuals that are in this case labeled as white Americans, individuals of European ancestry. For example, for the TNNT2 gene, only 2.9 individuals or 2.9% of individuals that are self-reporting as white Americans carry that mutations, whereas almost 1 in 3 individuals that are African Americans carry those mutations. And since this is a rare genetic disorder, this high frequency in African Americans is not consistent with the fact that this is supposed to be a rare disorder. And for sure, it's not the case that 1 in 3 individuals that are African Americans carry this cardiomyopathy. So what's the logical conclusion of this, and this is what this paper explored and proposes, is that this is a result of of this mutation not being pathogenic in African Americans. So it should not be used as diagnostic tool in this particular ancestry groups. And they draw the conclusion there. They actually go even further, and they sequence a bunch of individuals of African American ancestry, and they show that these individuals cannot, or these mutations could have been avoided to be used as for clinical testing in such groups. And this is not just one random example. This has been done throughout, and more papers are reporting these variants that have effect that are pathogenic just in some population groups, but not others. So in one of the more recent works from the EXACT consortium, they identified 163 of such variants that might actually need to be reclassified, or at least not used as diagnostic tools in certain population groups. So based on this idea, we took this thought and we took it into our data. So what we looked at is about 10,000 of these so-called pathogenic variants that we added on the genotyping array that's performed in Atlas. So we took all these 10,000 and we started filtering them based on a bunch of different criteria of QC, and then we asked whether these variants that are supposed to be very rare because they are impacting rare diseases, whether they are high frequency in some particular population groups. And I'm happy to talk more about it, but in the interest of time, here's two examples of such variants that have very high frequency in certain population groups. The first one is in people that self-report as Asians and not Hispanic. The second one in African American individuals that are also self-report as not Hispanic. And what you can see is that 15 to 20% of individuals that are within those population groups carry mutations that are supposed to be pathogenic in those population groups. But that's clearly inconsistent with those mutations being pathogenic and causing rare disease. So the easy explanation for this is that they are not pathogenic and they shouldn't be used for testing. So they're candidates for clinical reclassification. Obviously, this is not the final proof. We're waiting for the exome sequencing data, but we're already seeing, that our data can help with curating and reclassifying clinically actionable variants. Another thing that we might do is to ask ourselves whether, now that we're genotyping or we're getting genetic data from 150,000 of our patients, where we find enough patients that have mutations in genes that are supposed to be high effect size, that the CDC recommends that all incidental findings there or all the mutations should be reported to those patients because there's clinical actionability behind them. So there, we conducted this research question by which we took the 10 most clinically actionable genes from they're labeled as CDC10, from BRCA1 and 2 to hypercholesterolemia to another Lynch syndrome, another type of cancers. And all of these genes have the property that CDC recommends that any mutations that have been discovered in these should be reported back to the patient because there might be clinical utility. Obviously, in our data, we cannot report anything back because everything is de-identified, and there's the link between, and the consent is not allowing us to return any result. But we can ask, as a research question, we can ask, okay, how many of the patients that we've genotyped carry these mutations? How many of these patients we could have essentially have some maybe yield in terms of the genetics? Because remember, we are not consenting patients based on whether they have this mutation. We're just consenting virtually everyone that we can. So what we did, this is just a snapshot of how we looked at this data. We started from all the variants that we put on the genotyping platform. And when we did this was as of a year ago. We had 12,000 patients that were genotyped at that point in time. And long story short, we identified 29 of these patients that might have 19 variants that could have been in the, clinically useful in this gene. Again, there's no return of results, so there's no actual clinical utility at this stage. This is all a research question. So this is how those 29 patients look like. Most of them have mutations in BRCA1 or 2. There's some mutations in the other ones. And then we asked, again, from a research question, because we have the DDR phenotypic data for these 29 patients, we asked whether these patients actually developed the disease that actually is linked to their particular gene of interest. So for BRCA1 or 2, we looked at all sorts of cancers, hypercholesterolemia, we looked at hyperlipidemia, as well as hypercholesterolemia. Then for Lynch syndrome, we looked at essentially any types of cancers. And as you can imagine, the vast majority of these patients actually developed those cancers, 23 out of 29, with 6 of them not having so far records in our system of developing these diseases. So from the perspective of potential utility, you can imagine that the 6 would be the ones to follow through. And here's some examples, and I'll stop here. And you can imagine that this would be what could be done in the future as more variants or clinical utility might be discovered for mental illness. In the last couple of maybe 5-10 minutes, I'm going to talk to you about a different project by which we use the electronic health records as a discovery tool to identify potential patients that may be misdiagnosed, or patients that have not developed all the symptoms that will classify them into a rare disease. So essentially trying to do an algorithmic job of prioritizing or identifying patients that might have rare diseases and routing them towards, in this case, an autoimmune specialist. So we focused on a disease that it's an autoimmune disease. This is a common variable immunodeficiency disorder, CVID. And the reason why we focus on this is because this is a highly heterogeneous group of disorders, and patients that eventually get diagnosed with the disorders, typically they spend maybe 6 months to a year, if not more, in the hospital by looking from one clinic to the other one, because they have manifestations that are involving virtually all of the organs, anywhere from let's say problems in the ears, bronchitis, pneumonia, red blood cell counts, and so on and so forth, and enlarged lymph nodes. So in general, they spend quite a lot of time before they are referred to autoimmune specialists, and then they actually are diagnosed with this autoimmune immunodeficiency disorder. So our main goal, our main hypothesis here was, can we use the electronic health record data to find a way where we can identify these patients beforehand and then route them to an autoimmune specialist? Is there a signature in their electronic health record that will allow us to pull them out before they spend this couple months to a year in the hospital? So just to give you an idea of what the data looks like, the CVID roughly affects one in 25,000 to 50,000, so it's a rare disorder. And there's a significant delay, as I mentioned before. And as you can imagine, the sooner we capture them, the better the outcomes they have. And again, this is just results showing you that the sooner we catch them, the sooner we can provide them a diagnostic, the better outcomes they have. So how do we went about to come up with an algorithm, a computational algorithm that takes electronic health records and computes a score, if you will, a risk score for a particular patient to actually have CVID. So what we took the clinical manifestation of CVID, and then we converted them into Phecodes. So from the clinical manifestation of CVID that we can find in, let's say, external database like OMIM, we pulled out all their symptoms as well as other related outcomes. We pulled out all their lab values, and in this case, the most interesting lab, or the most predictive lab, I should say, is the IgG values. But this is a general lab that can be elevated due to various due to various other diseases or various other reasons. So it's not specific to CVID. So we pulled all these things together, and looks something like this. So if you go into the actual definition of the CVID, it has autosomal recessive, has all these features, all these symptoms that are involved, head and neck, respiratory, and so on and so forth. Each of these, we map them back to a Phecode, and you see here the list of Phecodes. There's maybe 15, 20 others. And these Phecodes, now we looked to come up with an algorithm that will aggregates all these Phecodes together into a single risk score. And in general, all these Phecodes, are associated to many patients in our system. And we're looking for a rare disorder. So we're looking in this CVID, we're looking at something that, a tiny fraction of a % of individuals or patients at UCLA have it. Whereas the Phecodes are common things. And in general, this is how it looks. So if you look at acute upper respiratory infections, roughly 13% of all our patients have this Phecode, whereas in CVID is 24%. Chronic sinusitis, it's overly enriched in CVID, but it's still 44% of all of our patients have. So in short, no single predictor does a great job, but when we add them all together, in combining all of them, we can identify roughly almost all the CVID patients that are in the system by putting this into a machine learning algorithm that will find the optimal weight from the data itself of how this Phecodes should be fitted together. And the way it looks is kind of similar to a machine learning algorithm. I'm happy to talk to you in more details, but you can imagine as we have a matrix of patients on the left there. Every patient has or does not have one of these Phecodes. And then some of these patients, we know by chart review and manual curation that actually have the CVID. So now we have this type of data where we can train a classifier. And in this case, we tested a bunch of them, but we focused on a machine learning tool called ElasticNet that incorporates and builds a risk score that puts different weights to these different Phecodes, and they're reflected here by the strength of this shaded color. For example, bronchiectasis, it has a much higher weight than other traits because it's more specific to CVID. And the nice part about machine learning here is that we don't need to restrict to Phecodes or billing codes, if you will, but you can start adding all sorts of features that might be relevant, and the classifier would put them together in the optimal way. So here we looked at lab values.
Just a note, you only got 5 minutes, Bogdan.
Okay, so I'll the classifier works better than not doing the classifier. This is showing you its performance. We see that within the top 1,000 patients out of 1 million, we capture 60-70% of all the cases that we've ever observed at UCLA. This is one example of one such patient where the classifier on the bottom, PhENet, is super elevated. So with the classifier, the risk score that comes from the classifier puts him at the 99th percentile, maybe like a year or a year and a half before it actually received a diagnosis of immunodeficiency. So we're optimistic that this score might be highly useful in practice. And now that we received or we're about to receive a grant from NIH, we're hoping that this would be actually useful in the clinic. Most of these patients have mutations that are related to immune deficiency. Again, super happy to talk more about it, but in the interest of time, let me wrap it up. So hopefully I convinced you that these two resources at UCLA, the DDR and Atlas, are extremely useful. And hopefully they'll provide useful for your research or for your questions that you might want to delve into. Hopefully I can convince you that although ICD codes are extremely noisy, by putting them in meaningful ways, we can extract useful information. The diversity in the genomes is extremely useful for a variety of questions, including fixing incorrect classifications. And hopefully I've convinced you that by building machine learning on top of VHR can prove to be extremely useful. Okay, so I'll stop here by acknowledging all the many people that have made all these resources available, from the Institute of Precision Health to departments such as computational medicine, such as the IT of the School of Medicine, and so on and so forth. This is just a snapshot of many of the people that have played key roles here, including on the IT side. This is an entire amount of work that has been developed for you guys to be able to utilize this technique, this resources. And ultimately, the folks to thank the most are the UCLA patients that have consented to be part of this research. And I'll stop here. Hopefully, there's still time for a few questions, Jonathan.
A couple of minutes left. So I think just one really important thing, Bogdan, if there's anyone in the audience who wants to take advantage of this resource, then who should they contact? Can you give us maybe a contact email or a website that they can go and look at?
Yes, absolutely. That's a great question. So if you go to the Institute of Precision Health website, there's all the information there about how you can, including email, as well as how you gain access to all these resources. So that's probably the best way to get started.
But I'm assuming if you are going to use these resources, most of us have no idea what a Phecode is or how to interpret it, let alone write some artificial intelligence algorithm to sort it out. I'm assuming you're going to have to do this in collaboration with someone like yourself in computational medicine who's a little bit more savvy around these issues. Is that correct?
Yes. So it depends on your expertise. Yes, you're absolutely right. So if you want to collaborate with any of us in computational medicine or IPH, please contact me. I can put you in a link with anyone with folks that are interested from the other side. We are in computational medicine are super interested in the machine learning component of it, so I can definitely put you in contact with anyone over there. If you have enough computational savvy to write some quick code to query these databases, everyone can get access. Everyone in DGC, SOM, and Semel can get access to this data and can provide some queries.
I think we're going to have to set up some coding classes if that's what's going to be needed. But anyway, so just before we go, there's one question. So two people have asked this. Really, it's about the how representative these biobanks are. One question was about the difference between those who did and didn't take part in it. So there's the difference between those who consented. And another question about the low minority representation, and is there anything going ahead to try and address these disparities?
Yeah, absolutely. So that's an ongoing task that IPH is doing. So there are multiple initiatives, which probably in the 1 minute I will do them a disservice, about increasing the representation in various different forms. Either on the genetic ancestry side, on the socioeconomic side. There are multiple different avenues for that IPH is seeking through, and I would say that if there are any ideas or any thoughts about how to do a better job or improve that from anyone this call, please reach out. It's kind of a group effort, because we definitely, IPH and all of us, want this to be as representative as possible. And in some cases it is, in some cases it could always be made better, and in many cases we don't even have a good metric of diversity for particular things, particular type of research question that some of us may be interested in. So yes, there are many ways. There's a lot to be improved. If there's any thoughts process on how to do it, please join us, or please email me and I can put you on.
Okay, thank you very much indeed, Bogdan. That's been really informative, and I'm sure you'll get a lot of interest after your talk. So I'll call the meeting to end now. We've gone a little late, but thanks very much for your attendance, and hope to see you all soon. Bye for now.
Thank you so much.